Bioxtyran Inc. They're an OTCQB company trading under the ticker symbol BIXT. The company is a clinical stage biotech with 3 revolutionary platform technologies that are quickly developed using the power of AI. They have a broad spectrum oral antiviral drug that recently completed clinical trials and achieved 100% viral reduction in 7 days, a feat only accomplished by one other drug in the past decade. Their drug is a galectin inhibitor that not only neutralizes COVID-19, but also neutralizes influenza and RSV and prevents it from entering cells. They're currently recruiting patients for their dose optimization study, which will give them the ability to proceed with their registrational trial in India after they finish. They have a second platform drug, an oxygenation molecule, being developed to treat stroke, and its development is supported by an FDA-approved device that detects local tissue oxygenation. Due to the molecule's small size, it has the ability to carry oxygen into areas of poor circulation. And the company has a third platform technology that's capable of treating cancer metastasis. So that's a lot. But before we get started with your presentation, I want to make a comment, Mike. You've been a guest on our show a number of times on our conference, and a lot of your focus has been viruses or your oxygenation molecule. But this time you're doing a whole presentation on cancer. So I have to ask, are you a little concerned that investors might view this as a distraction from your core technology?
Yes. That thought has crossed our minds that this may come across as a distraction. But the real target of this presentation includes big or little pharma, not just necessarily investors, okay? But here's the thing. We're using this conference to demonstrate our competency basically in landing a partnership. We actually know what we're talking about when it comes to cancer. After all, our CEO, David Platt, started out with cancer drug development about 30 years ago. What I'm presenting today is really just a teaser of what we think is one of the most thorough cancer presentations out there. We thought it was important for investors to see the compelling argument that we're going to be making to big and little pharma, and let them decide if our company now it's a $10 million market cap makes sense, while these other oncology companies who are going after this tiny patient population compared to what we're doing are worth billions.
Okay, so it sounds like you don't really view it as a distraction at all. So thank you for humoring me with my curiosity. Let's get down to business, Mike. I'll let you get started with your presentation and whenever you're ready, and let me know when you are ready for the Q&A.
All right, sounds good. So, we're going to do this presentation on galectin antagonists. Here's our forward-looking statements. Now, before I get started, I want investors to know that this cancer presentation that you're going to see today is actually a product of 16 years of research. Back in 2008, I witnessed the power of a galectin antagonist and how it brought a friend of mine with metastatic breast cancer back basically from the brink of death in 3 days. Then she was released from the hospital in about 3 weeks. I promised myself back then I would not let this technology die because it was just basically too important for humanity. Now I stand before you with a mountain of scientific evidence, not just one anecdotal story. And what we're saying, our approach to cancer, it needs to expand. We have to start targeting cancer metastasis. We have the technology right now to bring cancer to its knees. We just need these pharma players to just pay attention, or we need investors to basically ante up for this technology that could revolutionize cancer treatment. So with my little preamble, let's get to our outline of what we're going to be talking about today. All right, so our bodies have a competent immune system and we can get rid of cancer. But the problem is it tricks us in a bunch of ways, and we're going to go over this. And then there's all these tools that we have to fight cancer. And then we're going to propose the idea of a universal cancer target. And then we're going to go look at some clinical trials in using galectin antagonists in cancer and see how they compare. So you're going to see here the truth is that metastasis kills. It's not the primary tumor. 90% of deaths that are associated with cancer are basically due to the metastasis. Now, in the past, what we've done is we do surgery, chemo, radiation, CAR-Ts, ADCs, immunotherapy, all that sort of stuff. And what it's done is it does shrink the tumor, but it also extends survival. Now, what's interesting is that we think galectin therapy is perhaps more versatile than what we've seen in the past because it's going to start targeting metastasis and it works with all these other treatments out there. Now, galectins here are linked to many chronic diseases. And this slide here, or this graphic that you see before you, what you see is there's arrows up and down. There's a whole bunch of different types of galectins. They're a target in cancer, a major target in cancer. But there's 15 of them, and you'll see that there's different numbers that correlate to them, and they're upregulated, downregulated. This is a product of 30 years of research. 11,000 journal articles on just galectin. There's probably around 4,000 on cancer and galectins. And galectins end up being a key biomarker of chronic disease. But despite all this evidence, and this is 30 years of research, there's no galectin inhibitors approved yet. Now we're going to take a trip down memory lane. Like, how does cancer form? And then we got a little graphic there. You have normal cells, and then follow along on the right, you see abnormal cells develop, mutations, whatever, cancer starts. Then they multiply uncontrollably, that's cancer. And then it grows blood vessels, invades surrounding tissue, all that stuff. But did you know this fact? The human immune system is so efficient, it's designed to detect and eliminate these abnormal cells before they really get going and become cancerous. And there's a little tidbit for you. The human body is actually capable of curing cancer and does so multiple times in our lifetime. So with that, why do people get cancer? And what happens is we have an immune system, and if it's competent, it's going to stop cancer. And look at our tools that we have. Look at our soldiers, if you would. If you look on the left, surveillance and elimination, those are T cells, natural killer cells. And then we also have tumor infiltrating cells. This is our arsenal to defeat cancer. And here's the brutal reality. The body's in this never-ending fight against cancer. And despite it, all our efforts to prevent it with these tools, 1 in 5 people in our lifetime is going to be diagnosed with cancer. Why is that? Because of this. Cancer tricks our immune system. And we're going to go over the defense mechanisms, what they are, and then I'm going to give you an overview of it. More detail is going to be in our full slide deck. But anyhow, cancer deactivates the T cells with something called the galectin effect. It alters the genes, and you can see all the different mutations down below. It suppresses the immune system, and it also creates this hypoxic environment where cancer can grow without basically blood supply. So let's take a look at what I call T cell deactivation and cancer's cloaking mechanism. So you'll see there's going to be a little picture there on the right of an insurgent outpost. It might look like a military outpost. We're going to call it an insurgent outpost. They're the bad guys, right? And the good guys are our T cell army. But what happens is as soon as they hit that barbed wire fencing, they get stunned, okay? That's the T cell energy. So they're not killed, but they're basically stunned and they can't do much. But and it also sends a message and a signal to their buddies like, oh, we got to bring in reinforcements, okay? And that's basically the cloaking mechanism of the immune system. So these reinforcements are suppressor cells, and they're like, hey, this tumor doesn't exist. Don't come here, right? And now we're going to take a look at a little bit more detail on this galectin effect that I called it, and how they stun this T cells, if you would. All right, so on the left, you're going to see a a T cell that's basically covered in this plaque. That T cell is not operating correctly. And then to the right of it, you're going to see the cell that is operating correctly. And you see what happens in T cells, they need to get good adhesion to the cell. So cells are round, and then when I mean good adhesion, I mean they need to smoosh together like this. And then all these little surface lectins, they come over here to where the antigen was presented, and they form this little smush barrier, and then they penetrate through just like that graphic is showing, and they pump in their cytotoxins. Well, what happens is when the cells surrounded with this galectin plaque, this little guy over here and this little tentacle, they need to move over here. They can't get there because they got to go through all this galectin plaque. And then there's this article that talks about the reversal of T cell anergy. And you'll see what happens is if you introduce a galectin antagonist, what it does is basically rips off this galectin plaque, and then the T cell goes back into operating mode. So instead of being stunned, it gets back into the game and starts fighting again. You see, that's T cell anergy. So we looked at the defensive mechanism. Well, how about the offense of cancer? It's metastasis, angiogenesis. So metastasis is basically tumor escape. It goes into the bloodstream and then out of the bloodstream and builds a new tumor. Then angiogenesis, that's building the blood supply. And then hypoxic conditions, that's growing basically without a blood supply in a low pH. Well, let's focus on how metastasis works or happens. So in the graphic, you'll see in the upper left, there's this massive tumors there. Okay, well, they need to get into the blood. They need to get into the blood supply. So you see what they do? They squeeze through. That's B. And then they roll around. They pick up some stuff. They basically get sticky, and then they attach somewhere, and then they come out of the cell and then build a tumor, and then the vascularization. That's how cancer metastasizes. So galectin-3, which is something that we block, it makes the tumor cells sticky, basically. So what happens if you take away their stickiness? Probably less metastasis. What's another way that they are offensive? Well, angiogenesis. Angiogenesis is basically growing a blood supply and vascularization to the tumor. So now I have here in red, there's activation, proliferation. These are all the things that we're able to do. But now this graphic, maybe we didn't pump it up enough, but there's galectin-1, galectin-3, galectin-8, all those little Pac-Man in there. What I want you to understand is galectins are integral to angiogenesis. So blocking them kind of blocks that angiogenesis. And then the final point that I want to make there, if any of you ever heard of a drug called Avastin, it's a VEGF inhibitor. Now galectins and their impact on angiogenesis is upscale of that. That means we're probably going to have a more profound effect than even the existing VEGF inhibitors. So now, in the next part of the presentation, we talk about cancer treatments. So there's basically 4 right now, and we could think of a 5th, but we'll talk about that later. You can cut a person, that's surgery, chemo, you can radiation a person, or you can melt. And we're going to focus on this one, and that's basically the checkpoint inhibitors where they kind of melt the cancer away. But I'm going to look at a special case here. And we have a theory, or this theory in this peer-reviewed journal article, explains actually why checkpoint inhibitors aren't getting the response rates that we would hope that we would get. Like they're not a cure, a complete cure. And the theory is successful immunotherapy requires an activated immune system. So what happens here in this theory that revolves around galectins is if you look on the left and you look down, it's suppressed. And on the right, it's activated. And then, so what we call that is your basically immune system is, the tumor is cloaked from the immune system. And then you have these, the anti-PD-1 and the galectin inhibitor, and it becomes uncloaked. So this is a peer-reviewed journal article that basically indicates blocking galectin-3 restores normal function to the immune system which means it'll eventually melt it away. Remember, like in my beginning slide, how tumor forms? Our body, if we have a normal functioning tumor system, is going to eradicate it. So here's what we think the evolution of cancer treatment is. We think, let's go over it real fast. So the timeline. So back in the BC days, look, people were cutting tumors out. That's how they were getting rid of cancer. And then radiotherapy, and then chemotherapy, and then you see targeted therapy. And then we went to these checkpoint inhibitors that we have right now. And we think the next evolution is targeting cancer metastasis with these galectin inhibitors. Now, the thing that I want to point out is the evolving clinical endpoints. So first we were talking about reducing the tumor. And then now we're into this progression-free survival. You know, sometimes the treatment or the cure is worse than having the disease, right? And that was the idea of like, look, we need to make sure people live. Yeah, it's great, we shrunk the tumor, but the person died. So now we're looking at stopping metastasis, and liquid biopsies are probably really good indicator of that. So now let me summarize everything for you on the treatment spectrum. So what we have here is targeting the primary tumor is not the solution. Cancer metastasis is this large unmet medical need, and there's plenty of ways to treat cancer, and a lot of them are effective, and they have minimum side effects. But the leading cause of death is still cancer metastasis, and we think that's the missing puzzle piece in cancer treatment today. So now we're going to talk about a universal target. Well, a universal target in cancer would be great. Right now we have many targets for cancer, but a universal target would be great. Now, what would that look like? So in the upper right of this chart there, the ultimate cancer target spans multiple cancer indications. So multiple cancer indications, multiple disease stages, right? And relevant for all combination therapies that exist right now, correct? So now look at this peer-reviewed journal article that talks about the different galectins, right? They talk about the cancer type in the next column, all different types of cancer, right? They talk about what it does and the mechanism. So there's a whole bunch of galectins implicated in cancer. Galectins are responsible for this proliferation, growth, invasion, angiogenesis, which is the definition of metastasis. And galectins represent biomarkers for bad outcomes for treatment in cancer. I mean, this thing spells it out as plain as day that galectins are a universal target. But there's more. Let's get to the next slide. Galectins are the missing link in cancer. And galectin science is actually in the shadows. So we looked at PubMed, and there's the graphic there. There's around 4,000 journal articles written on galectins and cancer in the past 30 years. No approved drug. And there's only 10 galectin antagonists that have really been developed in the clinical stage. And what's interesting, anecdotal fact, is our CEO developed or invented about 6 of them. And there's no existing big pharma collaborations out there in galectin science that we know right now. And zero oncology trials are embracing this concept of high galectin-3 expression in these subgroups, despite the fact that it shows efficacy. We'll get into that later. All right, a lot of drugs are resistant to cancer treatment or become resistant to cancer treatment. I don't care if it's and look at the graphic, there's radiotherapy, there's targeted therapy, and there's chemotherapy. Again, this peer-reviewed article hits the nail right on the head. The resistance in cancer treatment is caused by these galectins. If you look at that graphic, galectins interfere with the process or are part of the process when it becomes resistant, right? So what happens if we block it? Galectins probably take away the resistance. So what this graphic or what this journal article is about is therapies, they're effective, but they're not sufficient. We can do better. There's room for improvement, and a galectin antagonist is that room for improvement. Now this is the money slide for the scientists in the audience. Galectin, there's many targets in cancer, and we've outlined 7 of them in this peer-reviewed journal article. But what we did is we overlaid that with research that shows how galectins are implicated in each one of these druggable targets. So it just makes, I mean, everything, it just makes sense that galectin should be used in combination with all these treatments. And there's the peer-reviewed research for the scientists to kind of look at there. And here's the thing that I want everyone, the investors should understand. Galectins are essential for the tumor's survival. You see that the graphic there explains everything. If you're a tumor cell and we take away your galectins, we're basically taking away your food, your water, your clothing, your shelter, and we're putting you out in the middle of the desert. What happens if we give you galectins? You thrive. The other thing that we saw about galectins is there's this thing called the galectin signature during diseases. And what happens is this galectin signature changes. And this is a case study on prostate cancer. And what we saw there is how it changes from basically from one disease state or one stage to the next, how you might start with high galectin 3 and end up with high galectin 1. Well, that's a great biomarker, isn't it? But instead of blocking galectins, how about we just treat them? Now, this is going to be a fun slide to look at here. Galectins impair the immune system, and there's a peer-reviewed article on it. But look at galectin. Look at the left. This is in pancreatic cancer. Do you want to be this guy with that's galectin-1 positive and only a 5% survival rate? Or do you want to be this guy that's galectin-1 negative with like between a 60 to 70% survival rate? Who do you want to be? So low stromal galectin-1 expression is associated with long-term survival in pancreatic cancer. Amazing. Now we're going to go over some clinical trial results with galectin inhibitors. High galectin expression leads to bad outcomes. This is a retroactive study of non-small cell lung cancer patients. And what they did is they did a biopsy of their galectin expression. If the red line means you had high, the green means you had low galectin expression. You want low galectin expression if you're going to do well on Keytruda. And as you can see, all the people in green responded. So that's 100%. So 100% of people responded there, and they had low galectin expression using Keytruda. And then almost 90% of the people basically that had high galectin expression didn't do well. So what happens if galectins were blocked? Well, we know the answer. And this peer-reviewed journal article, it's down here at the bottom there, shows abelapectin in cohort 2 had 100% objective response rate in advanced melanoma in an 85-day trial using the optimal dosage. There's a waterfall plot down there that basically shows you the complete responses. But I want you to compare Keytruda, and that Keytruda is 33% response rate. So that's that dotted green line below the threshold of what's called the progressive response. And you can kind of see it just barely met it, right? And that's basically 2 years. Now look at the data. 1 in 5 had a complete response. Complete response. Gone, right? Not just progressive, and basically 100% of the people had a progressive. So, you do the math, 100 versus 33. Is that good? But look at this picture. This picture's telling. Look at all the arrows on the guy. Those are tumors. After therapy, look at that. Completely clean. All tumors resolved. That was in that peer-reviewed journal article. So I want to highlight that. Let's look at another peer-reviewed journal article. Or actually, no, this is just data. GB122 in non-small cell lung cancer with ATZ, right? Look at this data here. These guys have tumor shrinkage 70%, 30%. What was it? 20. So they have shrinkage. Look at this response rate. It's 40% in just the first cohort. The second cohort was at 60%. This is pretty incredible data when you consider the objective response rate. Look in the lower left in non-small cell lung cancer is is 22 to 38%. So combination therapy makes a lot of sense here. But then there's one other point that we found out. Galectin-3 serum levels decreased in responders. So blocking galectins ended up in a good outcome, we could say. And then if you look at the red, those are bad outcomes. So if you weren't able to block them, you had a bad outcome. So anyhow, right now we got to wrap up. There's a whole bunch of galectin antagonists in clinical trials, and we're part of it. And anyhow, let's just end the discussion there. Thank you. Anna?
All right, Mike. Well, fascinating presentation as always. So after hearing, the first thing that pops in my head is you've identified the universal cancer target. Don't you think we'll find a cure for cancer?
The reality is that many cures for cancer basically exist right now, but they're functional cures. And what I mean by a functional cure is that if you can catch it early enough, you can cut it out, you can burn it out, you can blast it out with chemo, or melt it away with these targeted checkpoint inhibitors. Now, the real question I have for you is can we find a cure for cancer metastasis? The answer is, though, not if we keep going down the same road. On the clinical trial websites, there's over 20,000 studies on cancer metastasis, right? And less than 1% are looking at how to stop it. What I saw were trials that were observing how the drug combinations work on existing therapies, or there were other trials that were trying to use liquid biopsies as a diagnostic marker. Now, what they're trying to do has a lot of value, and I don't want to take away from that, because what they're trying to do is find out the predictive value of existing therapies, or basically how to monitor people to see if their cancer comes back. So, but what I demonstrated to myself with this exercise is the healthcare industry simply isn't focused on finding a cure for metastatic cancer. And there can be no pathway to a functional cure for cancer metastasis if no one's. We need to look at other options, and I think we're more or less alone in our quest to treat cancer metastasis, which means we're actually on the cutting edge of science. It's for that reason I hope investors can see that we have a solid plan to take on cancer metastasis, but we need to prove it, and that takes money, and that's why we need your support.
So in your presentation, you mentioned over 90% response rates with respect to blocking galectins in cancer. 90% sounds pretty good. So what's going on with your partnership discussions? Are you getting pharma's attention?
You know, we've gotten a little bit of feedback, but it's not on the topic of cancer because, hey, we're unveiling this now. What we've been pitching is antiviral or dermatological applications of our tech. What we found out is that other galectin antagonists that have had a readout have had these conflicting signals in Big Pharma's head, and that left Big Pharma with this no aha type moment for them to basically pay attention. Once we get our aha moment, if you would, in Big Pharma, I think the tenor of our discussions is radically going to change. What we need is a major positive readout on galectin antagonists that actually worked in a phase 3 trial. Now, those positive results are basically going to bring this whole sector a lot of awareness. It's been lacking in the past. So we're preparing we're preparing presentations similar to this cancer presentation in the hopes that a major readout happens in a clinical trial with a galectin antagonist. And the trial we're waiting for is supposed to read out at the end of the year. So we're hoping to get Big Pharma's attention basically when the new year comes.
So in your presentation, you mentioned oncology trials haven't embraced the concept of galectin-3 subgroups. So I'm trying to follow, how's that relevant to your work in cancer metastasis? Can you give us a better context of why this is important from your standpoint?
Well, yeah. Before I talk about the relevance of these subgroups, I need to just make a comment. If there's any biotech audience who owns stock in an oncology company that basically failed the clinical trial, like in immunotherapy, you need to listen up here, all right? Odds are your trial failed because they didn't factor in people with high galectin-3 on their tumor. And those tumors are suppressed by the immune system because of the galectin-3. If you want to make that money back, right, then you're going to need to convince management to run trials that segregate people out by the galectin-3 expression. The science has been out there for basically 5 years, yet the oncology companies aren't taking advantage of this almost certain pathway to approval. It's a fact that people with competent immune systems will do much better in cancer treatment than those people that are impaired. Now, here's how it's relevant. The top checkpoint inhibitors, Keytruda, Opdivo, and Tecentriq, these drugs are effective because they empower your immune system, but at best they work 40% of the time, which means they fail 60% of the time. These immuno-oncology drugs are virtually unchallenged because few basically competitors can get to these high response rates. That can change overnight if some of these drugs that showed little efficacy, but not enough to basically get over that finish line to the approval, go back to the drawing board and basically modify their inclusion criteria by galectin expression. Once a couple of drugs redo their studies and come back from basically the graveyard and show, hey, look, galectin-3 works in this subgroup, give us an approval, then we think things are going to change because once these drugs make it to market and start siphoning the sales off of big pharma, they're going to start to pay attention. They're going to find out that these galectin subgroups are responsible for that efficacy in that trial. And that's what we ultimately want. We want drug companies looking for galectin antagonists. Now, one final anecdote. What's amazing to me are these oncology drugs that failed their trial because the placebo group did better than the people taking the drug. Ever hear that? These companies developing these drugs spent all this money trying to justify how well the drug really worked, if not for this little aberration here or there, and then promise we'll do better next time. Well, the reality is that they probably got a number of people with low galectin expression in their control arm. What they should have done was categorize people by their galectin expression on their tumor, because that's what we learned in this presentation. Galectins impair the immune system and interfere with all the existing treatment options right now.
So, Mike, with time,
we good on time?
I'm going to go to that last question. You just said it. Galectins impair the immune system. So does everyone with cancer have an impaired immune system? And are there drugs in development that can correct the immune system?
If you have cancer, your immune system is impaired, and it's impaired for some time, right? Remember back to the presentation when I talked about the galectin effect. It's creating T cell energy in your body where the T cells simply aren't responding to the cancer. Remember that graphic with the boy in the middle of the desert with no food or water? I'd venture to say his activities were impaired, wouldn't you? Well, that's what cancer does to your immune system. What we need to do is turn the tables on cancer and impair the tumor by blocking galectins and withholding the things that it needs to survive. There's no drugs that I know of specifically designed to restore balance to the immune system like galectin inhibitors do. So I got to end on that note, I guess, right?
It's a good way to end. Mike, always fascinating listening and learning from you. Thank you for joining us again on the conference, and we look forward to seeing you real soon.
Yes, indeed. Thank you.